أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم لم يكن الذين كفروا من أهل الكتاب والمشركين منفكين حتى تأتيهم البينة رسول من الله يتلو صحفا مطهرة فيها كتب قيمة وما تفرق الذين أوتوا الكتاب إلا من بعد ما جاءتهم البينة وما أمروا إلا ليعبدوا الله مخلصين له الدين حنفاء ويقيموا الصلاة ويؤتوا الزكاة وذلك دين القيم صدق الله العلي العظيم. 16th lecture for the first year class, and I would like to tell you today the number of the chromosomes. How many number of the chromosomes are present in the human body? Chromosomes numbers matters because I was telling you in previous sessions that the variation is caused just because of different number of the chromosomes. If the chromosomes are different, number of the chromosomes are different, the variation will be caused, and the variation depends upon the DNA, hereditary material, the four type of the nucleotides. If you are watching a slide, you will see the chromosomes which is visible to you. I will label one by one the all parts of the chromosomes in the last session, but earlier I just want to tell you that chromosomes numbers are different in the different individuals. Some animals have 16 number of the chromosomes, some animals have 25 number of chromosomes. So different frog has different. Why frog is frog? Because it has different number of the chromosomes. Why humans are different from each other? Because humans have the different number of the chromosomes. And simply the DNA and the chromosomes are related with each other. How they are related with each other? Because in the previous sessions I was naming you two things which are causing the variation. One was the chromosome, one was the DNA. These both are causing the variation, and now I will confirm what is the relationship between the chromosomes and the DNA. If you will see this one, the diagram, I am just magnifying, it's visible to you, that is composed of the DNA. Chromosome is composed of the DNA, and the DNA consists of the four nucleotides. The relationship is that the composition of the chromosome is the DNA. The integral part of the chromosome is the DNA, and the two things which are present in the chromosomes, they are two in numbers. One is the histone, which is the positively charged, and one is the DNA. Out of these both things, the one thing which is very important, that is the DNA. The DNA is hereditary material. I will define once again what is the hereditary material. The material which is transmitted from the parent to the offspring, that is the hereditary material. If you will see the parts of the chromosomes, which you will see here, the slides, the central part of the chromosomes is important because it consists of a protein which is a kinetochore. Now I have to label that kinetochore. Yes, I will tell you now. You can see the central part of the chromosome, that is the kinetochore, and this protein also I was mentioning present in the spindle fibers as well. And I would like to tell you, I would like to share one information, that chromosome attach with the spindle fiber just because of the kinetochore, because kinetochore is a specific protein which attach the chromosome with the spindle fibers. So like dissolves like, the kinetochore attached with the kinetochore. Now there are two type of the constrictions which I would like to tell you, which is the part of the chromosomes. Two things I am telling you, that the chromosomes cause the variation, the DNA cause the variation. And one thing I am telling you, the central part of the chromosome is the centromere. Central part of the chromosome is the centromere. And centromere consists of a specific protein, special protein, which I have highlighted now. You can see this one with red indicator, that is composed of kinetochore. And kinetochore is a specific protein that attach with the spindle fibers. And spindle fiber attachment is so much important during the cell division, because kinetochore will not attach with the kinetochore, chromosomes will not attach with the spindle fiber, no cell division will take place, no increase in the size will take place, and no even variation will take place. So now, number of the chromosomes, this is a question. What is the number of the chromosomes in the humans? 23 pairs. I am not telling you 23 number of the chromosomes, I am telling you 46 number of the chromosomes. So total number of the pairs which are present in the human body, those are the 46 number of the chromosomes, and they are the 23 pair of the chromosomes, and they are arranged in such a way in the nucleus that the larger size, then smaller size, then smaller size, and last one is the sex chromosomes. And this arrangement of the chromosome I will teach you in the next session, in the next year, I think so. I think so in the next year I will tell you that arrangement of the chromosome in the nucleus is called as the karyotype, K-A-R-Y-O-T-Y-E-P. I am indicating karyotype is not important for you because you know that you will study in the next session. The point I would like to tell you, that is very important, the central part of the chromosome, exactly the central part of the chromosome, which is important. This is composed of a specific protein molecule that attach with a specific spindle fiber, and spindle fiber, you know, that is formed during the cell division, during the mitosis, during the meiosis. You remember that mitosis, meiosis, 2 centriole were shifting towards the right side, 2 centriole were shifting towards the left side. What was happening? Between them, spindle fibers were being formed, and this indicator is telling you, this part is telling you, this circular part is telling you it consists of specific protein which is kinetochore. So variation, why the variation is caused? Because chromosome consists of different sequence of the nucleotides in the DNA. I am different from you because I have the different sequence of the DNA. If you are different from your father, because you have the different sequence of the DNA. The 4 types of the nucleotides which are present in the DNA, one is adenine, A-D-E-N-I-N-E, adenine, thymine, guanine, cytosine, the 4 types of the nucleotides. And these 4 types of the nucleotides will carry the character of each individual. It means that whatever your internal things are present, these are just due to sequence of the DNA. And if some characters are similar to the parents, the reason is that because some characters are similar to the parents, reason is that because DNA is coming from the sperm and the egg, which is coming from the father and the mother. Some relationship in the sperm, some sequences in the sperm, some sequences in the egg will remain the same, and they will not change because mostly will change. Reason because they will combine to form the zygote. This central part, kinetochore. Now I will tell you the secondary constriction, the primary constriction. These are the 2 new words which I would like to tell you. The primary constriction, which is present at the center of the chromosome, that is called a centromere. I am using the word centromere. Central part, centromere. Composition of centromere, once again the same thing, kinetochore, K-I-N-E-T-O-C-H-O-R-E. Look here, if chromosomes occurs in the form of the pairs, and total 23 pairs of the chromosomes are present in the nucleus, and chromosomes are important. DNA is important because chromosome is composed of the DNA, and the DNA is hereditary material due to sequence of the DNA. What is the DNA? Long lengths of the sequence of nucleotide: adenine, thymine, guanine, cytosine, thymine. This is the sequence of one individual. Another sequence of another individual: adenine, thymine, guanine, cytosine. Even one nucleotide sequence will change, the character will change. So DNA of each million, trillions of the people, that is different just because of sequence of DNA. Now the significant thing which you have a question in the mind, so why the histone is present in the chromosome? Because chromosome is composed of the 2 types of the structures. One is the DNA, one is the histone. And what is the need of histone? To be the part of the chromosomes. Yes. Another unique thing about the histone is that histone is positively charged. Histone is positively charged. The positively charged particles present in the DNA, just because of the scene, the positively charged particles, they are present in the DNA. The reason is that because the DNA has the negative charge. And negative charge combined with the positive charge, and then the DNA will be able to bond with histone. Histones are small balls-like structures. There are many balls in the nucleus. Reason is that the long lengths of the DNA, they cannot be present in the nucleus due to the shorter space. If you want to increase the space of the DNA, how you will do? These small balls will just roll the threads of the DNA upon in such a way that they will form the conjugated molecule. And conjugated molecule, you know, that is a nucleosome, that's the conjugated molecule. Now I would like to tell you the primary constriction, secondary constriction. These are the parts of the chromosomes. I will speak slowly now because I am going to tell you the name of the parts of the chromosomes. The primary constriction now, kinetochore, clear. The secondary constriction, yes, this is the area which will form the nucleolus. Do you know, in the last session I was telling you one organelle which is without membrane present in the nucleoplasm, producing 3 types of RNA: messenger RNA, transfer RNA, ribosomal RNA. These 3 types of the RNA which are found in the nucleus, and ribosomal RNA was present in ribosome. It was forming the protein. The transfer RNA were forming the amino acid. The messenger RNA was getting the message from the DNA in the transcription, and that was going to the ribosome. to form the protein in a translation. 3 types of RNA, messenger RNA I explained, remaining RNAs I will explain in the next session. So chromosomes, once again our headline is the chromosome, because that chromosome changes the life of the people. If the chromosome sequence changes, the disease occur. If the chromosome sequence changes, the cancer occur. Chromosome sequence changes, the character changes. The DNA, the mutation, which is the sudden change in genetic material due to the chromosomes, and each factor in the life, even the suicidal act of the cells, even the autophagy, even the digestion in the body, even the circulatory system, even everything is being controlled by the chromosomes, and chromosome is being controlled by the DNA. Histone need was just only to bound the DNA in the little space. So you got the idea. This structure which is visible to you, two threads which are coinciding, which are combining in the center at a specific part. You can see a specific part. This is the centromere. And centromere is also called primary constriction. It is composed of a protein, kinetochore, and kinetochore is the protein which will attach the chromosome at the spindle fibers during the cell division. And now we go back to the next type of the centromere. Next type of the centromere, or you can say the secondary constriction. Yes, this one. Students, this is a new word for you, the secondary constriction in the chromosomes. Reason, because this is the area which will form, which is the nucleolar region. Nucleolar region, because it will form the nucleolus. Plural of nucleolus is a nucleoli. It will form 3 types of RNA. The one type of RNA I have explained, that is messenger RNA, that is a transfer RNA, that is a ribosomal RNA. The transfer RNA is very important because that it will combine to form the amino acid. The messenger RNA is also very important because that will get the message from the DNA from the nucleus in that transcription. And then messenger RNA will come through endoplasmic reticulum to the cytoplasm. What will happen? You know, messenger RNA will go to ribosomes. It will get the message. It will give the message to ribosomes. Ribosomes will call the amino acids by the transfer RNA. The different type of amino acids will come. They will be arranged in a sequence according to DNA, and the specific protein will be formed in a translation process. These all processes you have to study in detail in the future. We'll go back to secondary constriction. The secondary constriction, I would like to tell you, this is the secondary constriction. Secondary constriction is a nucleolar area. You are watching another area behind the secondary constriction. If you are watching here, you are watching the area at the just at the behind of the secondary constriction. This area is called the satellite, satellite. The satellite is important in the sense because it consists of useless sequence of DNA. Until now, I was telling you that sequence of the nucleotides in the DNA is significantly, substantially important. But I am telling you a part of the chromosome, which is the satellite, which is situated at closer to the secondary constriction. This part which is available near the secondary constriction, it consists of junk DNA. I will show you a slide after some time and you will be able to understand that what is the junk DNA. Useless sequence of DNA. It means I am starting from here, from this sequence, the secondary constriction. I will go from here to here. Secondary constriction, primary constriction. This is the secondary constriction up to here, from there to here. The sequence of the DNA. All are useful, but when you move from secondary constriction to the telomere, I used another word, the telomere, I have to explain now, end of the chromosome. The end part of the chromosome, that is called as a telomere. That is the end part of the chromosomes, and end part of the chromosomes are so important because they will not combine 2 chromatids. What is the chromatid? What is the chromatid? Yes, one chromosome consists of 2 parts. One is this chromatid, second one is this chromatid. I will teach you in interphase why there was need of 2 chromatids. The end of the chromatid is called the telomere. This is telomere, this is a telomere. So one telomere, the next telomere. The reason of the telomere is that they will repel. They have specific charges and they will repel each other. And benefit will be that both parts will not combine and they will repel. So they will prevent the combination of the 2 chromatid at the corners. So this is clear now. The satellite, once again I have to speak about the satellite. That is the junk DNA. That is the useless sequence of the DNA, and the useful sequence of the DNA start from secondary constriction. I am labeling now. Okay, clear? Secondary constriction. We go to this one, to the primary constriction. Primary constriction is also called as, that is visible to you, I think so. Primary constriction is also called as the centromere, and centromere consists of specific protein kinetochore. It goes from primary constriction to the secondary constriction. Now the secondary constriction is a nuclear region, and then from secondary constriction, you can see now, I am just highlighting the secondary constriction to the secondary constriction, that is consist of useful sequence of the DNA. That will not consist of the junk DNA. Junk DNA will be at back side of secondary constriction. Look here, I start from secondary constriction, it will go to this one, and when it will go to this one, that is the secondary constriction. At the back side of secondary constriction up to the telomere, this part is the satellite, SATELLITE, useless DNA part. So chromosomes has two parts. One is the part with the useful sequence of the DNA. Why it is called the useful sequence of the DNA? It consists of the body message. Useless sequence of the DNA, it never mean there is no function. There is no function. That is just a sequence of nucleotide without the message, and this message will not be carried to messenger RNA during the transcription, and this message will not be translated to formation of the protein in the translation. So end part of the chromosome will be called as the telomeres, and telomeres will repel each other. I am just revising. That will repel each other in such a way that both chromatids will not combine. Now I just want to show you some points which are very important. The telomeres, I explained the telomeres now. Telomeres you are watching now, this is the telomere. You are watching the satellite as well. Okay, I will just open the area, that is the satellite. I will highlight now, just hold on. This is the satellite, you can see. Okay, this is the telomeres. Meaning of satellite, the useless sequence of the DNA. Meaning of the telomere, to prevent the combination of the end of the chromosomes. Two words I have to explain now, euchromatin, heterochromatin. You know, I am telling you karyotype arrangement of the chromosomes in the nucleus is called the karyotype. There is question comes in the mind, how these chromosomes, we scientists were able to see that first pair, second pair, third pair, fourth pair, fifth pair, up to 23rd pair of the chromosome. How they know karyotype, how they understand that? They understand by staining the chromosomes. They use specific chemicals, colored chemicals, and many stains were used. Some stains were effective for some part of the chromosome. Some stains were effective for other parts of the chromosomes. This effectiveness of the stain on the chromosome divided the chromosome into two types. Now I will tell you the two types of the staining procedures, because those will be very important. Because if you are a scientist and you are just checking the chromosomes by using the chromatins, by watching the chromosomes, that is euchromatin is clearly visible. And it is lighter in color. Even you are staining, it is not dark, and it is clearly visible even without staining. Little a bit part of the visible. This type of the chromosome will be called as a euchromatin, EUCHROMATIN, euchromatin. And some part, when you will stain, different stains were used. I am not using the name of the stains because many stains are used. I will use two stains in the next chapters. Now I will tell you, when you will use the stains, like acetylamine, or you can say there are some other stains as well, because I will use the stain according to the practicals which you will use. That's why you will not be confused, because I may not use the stain which is not being used in the practical. Okay, heterochromatin. When you will use the stain, that is the black color, dark color will explain the part of the chromosome which became dark when you will stain, like acetylamine, acetylcholine. You will use the stain to color the chromosomes. The chromosome becomes black, chromosomes becomes dark. Such chromosomes are called heterochromatin, HETEROCHROMATIN, heterochromatin. So it means that on the basis of the staining, you can divide the chromosome into two types, the euchromatin and the heterochromatin. We'll go back to some of the details which I just want to share some information, which are so now I can tell you. One point I forgot to tell you. During the cell division, what is happening to chromosomes? Do you guess? Do you think yourself that what happens to chromosome during the cell division? Yeah, during the cell division, chromosomes were visible or not visible? Or without cell division, what is the fate of the chromosome? It was visible or not visible? I told you in the junior classes what was happening. chromosomes were in the form of thread-like structure. They did not look like chromosomes because they were not condensed, they was not thick, they were just thin, they were just in the form of the fibers, and they were not visible. They were just in the form of the chromatin network. When they were in the form of the chromatin network, they were in the form of the chromatin network when the chromosomes are not dividing. Mean in the normal cell is not undergoing mitosis, cell is not undergoing meiosis. What will happen? The chromosomes will be in the form of chromatin network. So when chromosomes will be visible, when they will be visible, they will be visible only when chromosomes will be visible only during cell division. During the mitosis, during the meiosis, chromosomes will be thick, they will be condensed, they will be visible, and when they will be visible, you can see, and you can, as a scientist, you can see, you can stain, you can see the number of the chromosomes, spindle fibers will be formed. So during cell division, chromosomes are visible. When cell division is not occurring, chromosomes are not visible. Now you are watching a slide. Satellite consists of useless sequence of the DNA. This is the definition I was talking about. You can see the slide. Satellite can consist of useless sequence of DNA, and that is called the junk DNA. Junk DNA means the useless sequence of the DNA. We'll go back to another slide. Primary construction is composed of kinetochore. I already told you. Kinetochore is important protein. What is important protein? I told you, which is present in the chromosome. Another protein which is positively charged. This is my quiz from my side to you. Do you remember the name of the protein which is positively charged? Do you know the name of the protein which is a positively charged? Yes, you have to tell me. Histone. If you will see this one, the primary construction is composed of the kinetochore. The secondary construction is composed of nucleolar region that will form nucleolus, that will form three types of RNA. And kinetochore is not positively charged. The positively charged protein is histone, that is part of chromosome. So if anybody say chromosome is conjugated molecule, what is the meaning of conjugated molecule? Two organic molecules, they are combined, and they are combined in such a way that they look like one molecule. That is example of conjugated molecule. So chromosome is a conjugated molecule. How two organic molecules combines? One is the DNA, one is the histone. One is positively charged histone, one is the DNA. They both will combine. The DNA and histone will combine. They will form the nucleohistone, nucleosome, and they both will combine in such a fashion that conjugated molecule will be formed. The last slide of today, which I will tell you in lecture number 16 for first year class, and lighter color with the stain, euchromatin. Clear? When you will use the stain, they are just become the lighter in color. They were not visible, and they visible only when they will be visible only the part of the chromosome which is visible with the stain, that is the dark. They become little dark, the visible dark, visible light, the euchromatin, visible dark by the stain, that is the heterochromatin. Useless sequence of the DNA, my quiz from my side to you. What is the useless sequence of the DNA? That is the junk DNA. That is the junk DNA. We'll go back to some informations. Once again, just want to tell you, satellite I told you, junk DNA I have told you, primary construction I've told you, secondary construction I have told you. One thing you remember, chromosomes are not visible. If you want to see the chromosomes, let the cell divide. Cell will not divide, you are unable to see. So if you are a biologist, you just want to see these all parts, you want to see conjugated molecule inside chromosome, you want to see sequence of DNA, you want to stain the chromosomes, you want to see telomere, you want to see satellite, you want to see secondary constriction, you want to see primary constriction. What you have to do, you have to wait for the cell division, because during cell division, chromatin network thread-like structure will become thick and they will be visible. I hope you will revise this one, and I will come back to you with the lecture number 16 very soon. Thank you very much.